Welcome to Monobiology. Today we're going to learn a very basic version, because we're going to do a more complicated version later on, but a very basic version of how DNA replication works. As we've talked about in the past, DNA replication is required before mitosis or meiosis can occur. In other words, in order to pass on the DNA from one cell to the next, you have to make extra copies of it, so that the exact same amount of DNA and type of DNA can be passed on from one cell to another. It happens in the cell cycle during the synthesis phase, or the S phase. There are 3 steps, if you will, to DNA replication. And you guys are going to take notes under each of these steps. The first one is initiation. But before I let you know anything about initiation, I just want to make sure that we're clear. Initiation, elongation, and termination are the 3 steps, and it's very important that you leave space, because I'm only going over the very, very basics of each of these steps. You are going to add to your notes after watching an animation about DNA replication. We're going to add more to it. So it's very important that you leave space between each of these. Probably, just to be safe, we'll say at least a third to half of a page, so you can add things that you need to add. Alright, so let's start with the basic concept of DNA replication. With initiation. Initiation is the beginning. It's the beginning part of the process. Now we know from past videos, or from past learning, that DNA is a double helix. It's a double-stranded piece of DNA that's connected in the middle via hydrogen bonds. Now that's kind of cool, because hydrogen bonds, although they're fairly strong, can be easily broken as well. And that's important, because what's going to happen in initiation is that this enzyme here, DNA helicase, is going to move along the DNA double strand, and it's actually going to unzip it. Okay, so the enzyme that does that is DNA helicase. It unzips the double strand. Now in this picture, it looks like the entire strand of DNA has come unzipped. But in reality, this happens at several locations throughout the double-stranded DNA. And again, that will be the more detail you get in the next animation that you'll watch. Okay, so that's initiation. DNA helicase separates the 2 strands of DNA by breaking the hydrogen bonds, essentially unzipping the double strand. The next step is elongation. To elongate means to make longer. So essentially what we're doing is we're building a new strand of DNA. Note that we have 2 dark blue strands. Those are the parental strands. Those are the strands that we started with. Okay, the dark blue strands. We also have these new, what we call nucleotides, which are the building blocks of DNA, that are being attached to the old strands. Okay, these are called the new strands, or the child strands. The child strands. Now, the enzyme that controls the building of these new child strands is called DNA polymerase. And DNA polymerase makes sure that the correct nucleotide matches up with the previous nucleotide. So notice here, we have an A. Well, what matches up with A? From our previous learning, we know that a T matches up with A. So DNA polymerase has to make sure that it brings over the correct nucleotide and attaches it to that A. Now, where does it get these nucleotides? Remember, all of this is taking place within the nucleus. And inside the nucleus, there are several of these nucleotides just kind of hanging out all around the DNA. The DNA polymerase is going to go ahead and pick up those nucleotides and bring them over to where the DNA replication is taking place. And it's going to make sure it matches up the correct nucleotide with its complementary base pair. And that's going to continue going along the separated DNA strands. Finally, we have the last part of this, and that's called termination. Termination is actually fairly complicated, but we're going to keep it really simple here. Okay, termination is essentially when both strands of DNA have been, both of the parental strands of DNA have been completely separated from each other and are now matched with complementary child strands. Okay, once that happens, once the DNA is completely separated and all the DNA has been matched up, the process is complete and termination occurs. And that's really it for this part. It gets more complicated when we look at the animation. The entire process of DNA replication is said to be semi-conservative. Now what the heck does that mean? Well, if we think of our original picture of our double-stranded DNA, this is the original piece of DNA that we wanted to replicate, and we look at our final picture, we can see what this concept means. Recall that these are the original parental strands. As you can see, in these new replicated DNA, each one of the new strands of DNA, the new double strands of DNA, has one parental strand attached to it and one child strand attached to it. Okay? Originally, it was thought that perhaps this strand was used to make a completely new double strand. That would have been not conservative at all. Or sorry, fully conservative. Or, in other ways, maybe this whole thing was broken up and each individual nucleotide was made to make new strands, and that would have been just ridiculous. But this is what happens. Each of these strands, these parental strands, let's just undo some of that, there we go, each of these parental strands, this one and this one, are used as templates, or if you prefer, blueprints, to build the new strands, thus making the process semi-conservative. Okay, here I drew up my own little version, just using the letters as opposed to putting the big pictures. And as you can see, this was our original strand. I did it in black, and to show you that the black ones are the original parental strands, and these, with their complementary base pairs, are my new strands, my new child strands. This is now the replicated DNA, and just to make a connection to what we had before, so imagine we have chromosomes, okay, so this is what's being replicated. Remember, we talked about chromosomes in meiosis. Originally, the chromosomes would have looked like this. Now this would be a double-stranded piece of DNA. At the end of the process, we're going to end up with this, which is now two completely identical strands of DNA. And that is what is going to form our chromosomes that are going to then go into meiosis and mitosis and be passed on to future generations of cells. And that in a nutshell is DNA replication. Make sure you've taken good notes on this. Bring your questions to class, but very important, leave space because we're going to add to this. This is just the basics.